right. Hi, everybody. I'm usually not this technologically challenged, but today I guess I am. So I'm Jillian Swinford. I'm a biologist with the invertebrate team. And so I guess you're all probably wondering, why I'm here talking to you about alligator gar. And that is because prior to this, I worked at Texas Parks and Wildlife in a applied research lab that primarily focused on um, fisheries, ecology, life history, as well as genetics. So we worked on a variety of different species, including invertebrates, but I also got the chance to work on fish as well. Um, and so that's what led me to working with alligator gar. And so this presentation is preliminary research from this project or preliminary data from this project. So I'd just like to, go through the work that we're doing down there. And if y'all have any suggestions as far as data analysis, I am all ears, but hopefully this is a nice holiday treat for y'all before, we head off for the holidays. So why do we care about alligator gar in Texas? Well, it is a species of concern in the region, and it is one of the last refuges of the species. And several major freshwater rivers in Texas support a large population and a thriving trophy fishery, which I will talk about a little bit later. So while gar are generally considered freshwater fish, they have become a major apex predator in estuarine ecosystems. Notably, their CPUE has been increasing in coastal regions in recent decades based on the TPWD gillnet surveys that we do annually. We've been doing since the 80s. So much more so that alligator gar are more abundant in Texas bay systems than other big marine predators such as bull sharks, other shark species. And I can attest to that from personal experience. The days are chock full of alligator gar. So these coastal gar are extremely prevalent, but their population and habitat use in this area is relatively understudied. And so that is why coastal fisheries got involved in alligator gar research. So y'all don't have alligator gar here in Massachusetts. So let's go over a little bit of the species, the ecology, life history, that kind of thing. So we know that, or many of us know that alligator gar are primitive fish, meaning that they're non-teleost fish. So they have a lot of unique adaptations, including things like they're able to gulp air, they're able to survive a wide variety of environmental factors. And they've been in the fossil record for 100 million years, meaning that they've been around since the Cretaceous. So this is an extremely old species of fish. They are long-lived, highly mobile, and pretty large. Right now, the biggest fish can get up to 10 feet. They might have been bigger historically. So this is a big animal. They're generalist predators that dominate southeastern river systems where they are still present, particularly in Texas and Louisiana. So they're the top predators in a lot of these systems. They're also particularly long-lived. For a fish, females can reach up to 50 years of age. And their movement is highly variable. They are highly mobile. They can travel up to 100 miles a year, but movement is, again, tied to seasons, particularly spring, in which reproduction occurs. So reproduction with alligator gar is highly variable as it is completely dependent on floodplain access. This means reproduction is tied to precipitation, so like periods of river flooding, as they deposit their eggs in these flooded tributaries on, vegetation to protect them from potential predators. So they need that floodplain access to be able to reproduce. Reproduction is also limited by a long time to maturity, which ranges from 5 to 10 years. So this is a fish that can grow to quite a large size and still not have reproduced. So while reproduction has been shown to only be in freshwater. Juvenile and adult alligator gar have a relatively high salinity tolerance. And that is up to 30 parts per thousand, though just from observations, it could be even higher than that. And fish in particularly that inhabit the lower sections of rivers more regularly access these estuarine ecosystems that have these high salinities. In addition, there may be some fish that are estuary resident for part or potentially throughout their entire lives, as evidenced by otolith microchemistry from a study that we did in 2017. So let's talk about a little bit about how they were managed prior to now. So historically, gar have been managed as trash fish via eradication. They have widely been considered pest or monster fish that greatly impacted important recreational and commercial fisheries as predators. And this eradication was pretty widespread in the Southeast and as thorough as possible. I've seen black and white photos of giant electrofishing boats built in the 50s and the 60s for this exact purpose, to eliminate alligator gar. And some of you might have a question, well, why don't we eat this fish if there's so many of them, they're so big? While you can eat them, and they do taste pretty good, they're like a mild white fish, they are so hard to process because of those really intense ganoid scales that cover their entire body. And these scales are so sharp that when you pick them up, they cut you. So a lot of people don't bother, and therefore it has become a trash fish. So because of this, the alligator's gar range has diminished from much of its historic range in the United States and Mexico. If you look at this map down here, the green areas are the areas in which the populations are still reproducing. Orange are remnant populations, so not reproducing, but still present. Yellow are the stocked areas as we're starting to build a trophy fishery. And then red is where they have been completely eliminated from the systems. So you can see the major states that still have big populations that are reproducing of the species are primarily Louisiana, Arkansas, and the biggest one is in Texas. So in recent decades, Texas has instated a trophy fishery, in part due to shows like River Monsters, which has encouraged anglers to come from outside of Texas to target these impressive animals, and also due to an extensive bow fishery that already existed. So this trophy fishery has allowed TPWD to bolster the state as a population refuge, and Texas contains the largest reproducing population of alligator gar in the country and I guess the world. So due to this interest, data acquisition and species management has become one of TPWD's top priorities for this species. So TPWD's goal is to better understand and better manage the Texas population of alligator gar, with the goal to support the trophy fishery, but also to support a healthy predator population of this very unique species. Most of the research up until this point has been done in freshwater environments and our major rivers and tributaries. So the focus of the research now is shifting to this group on the coast. As I've mentioned before, transient and estuary resident individuals are understudied in the state. So our goals with this project are to understand gar movement between these habitats, whether it's, seasonally, or whether there are other factors influencing, this movement. And we're going to do that primarily by using acoustic telemetry, which is fun when you have a fish that's completely covered by super sharp scales. And the other component of this study is to understand the age structure of coastal alligator gar, to see if fish are using estuaries at different times in their lives, or if they're present, there's a contingency that's present, throughout their life. But for today, we're just going to be talking about the movement study. So before we get into this, I'd like to introduce y'all to the Texas Gulf Coast. As many of you may not be familiar with the ins and outs of this area. So Texas Gulf Coast is huge. It is 367 miles of coastline. stretching all the way from the Louisiana coast or the Louisiana border to the north to Mexico in the south. And it takes 8 hours to drive from Brownsville all the way up to Port Arthur. I know because I've done it. So this area is primarily dominated by barrier islands that enclose extremely shallow bays. And these bays are under 1 to 2 meters. Oh, there we go. In depth in most areas. So we're talking very, very shallow, very different than what we see here, save for the channels that have been dredged out. So these barrier islands, unlike the East Coast, are largely underdeveloped, save for a few metropolitan areas like Galveston or Corpus Christi. Most of these Texas barrier islands are uninhabited and only accessible by boat, which is really unique and really interesting. These barrier islands protect 7 major bay systems. We'll go north to south. We have Sabine Lake up by the Louisiana border. Next is Galveston Bay. That is where Houston is. And Matagorda Bay, which is where I was located. And those are our 2 biggest bay systems on the Texas Gulf Coast. Then we have San Antonio Bay, Aransas Bay, Corpus Christi Bay, followed by the Laguna Madre. The Laguna Madre is really unique because it is a hypersaline lagoon, meaning that in the summertime, salinities can reach up to 60 parts per thousand, which is really interesting. There's a lot of interesting things going on ecologically here. If you have questions about the Laguna Madre, feel free to ask later. It's very interesting. There's not a lot of alligator gar down there, so we're not going to talk about it today. So these areas are dominated by large areas of marsh, small mangrove forests, and freshwater swamps. And primary structure-based habitat in the bays are oyster reefs and seagrass beds. Oyster reefs to the north and seagrass beds to the south. The area in which our study is going to take place is this small section of coastline between the 2 largest bay systems, Matagorda Bay and Galveston Bay, and it is about 18 miles in length on the coast. So in addition, the coast also serves as a drainage for several major rivers. The only one y'all would probably be familiar with would be the Rio Grande. For this project, our study contained the mouth of the Brazos River, which is shown here, which is a large river system that cuts 1,280 miles across the state. So it is a huge river system with ample habitat for alligator gar. The other interesting thing about a lot of these rivers is that they have heavily modified deltas. You don't see that, usual spread out delta when it meets the Gulf of Mexico. They've been highly channelized, and most of that sediment gets dumped right into the Gulf of Mexico. In addition, the last aspect of the Texas Gulf Coast that is unique is the Intercoastal Waterway, or the ICW, which some of you may have heard of. The Intercoastal Waterway is a large channel that was dug primarily for shipping and barge traffic along the coast. There is a northern component that goes from to Chesapeake Bay to Florida, and then a Gulf portion that stretches from Florida to Texas. The Texas portion stretches down the entire coastline and is often the deepest part of the estuary because it has to be dredged out for the ships. So first, let's talk about the study site. Cedar Lakes, now that we got that tour. So this is our smallest area in the coast that the TPWD Coastal Fisheries Long-Term Monitoring System samples. It's comprised of several small marsh lagoons situated along the ICW, which, as you can see, runs all the way down the coast there, between Galveston and Matagorda Bay. Galveston Bay is to the north, Matagorda Bay to the south. In addition, the Brazos River connects with the ICW here and enters the Gulf of Mexico here. And that was that really long, over 1,000-mile river that I mentioned earlier, which is a major highway for fish passage, has lots of habitat. We also have the San Bernard River, which is a shorter and smaller river, but still contains ample habitat for the species. And then we also have the San Bernard Wildlife Refuge down there, which is a huge area of marsh and tidal habitat. And then we have Freeport, Texas to the north, which is highly industrialized with oil and shipping industries. So we have a lot going on in this one area. But you can see this area serves as an interesting network of bottlenecks to fish movement. So the red diamonds mark the receiver sites, which we prioritized on the ICW, on the rivers and on the marsh lagoons. And then the blue circle indicate our catch and release sites. This circle area right here is what we will be calling from this point forth the Cedar Lakes Marsh Complex, which contains a lot of the habitat that we would expect gar to want to be using. Another factor of this area, interestingly enough, is that it currently has the highest gillnet CPUE for alligator gar on the Texas Gulf Coast. And this is based on our annual resource surveys that we do every spring and fall. So this figure shows gator gar CPUE from all the major areas sampled by TPWD on the Texas Gulf Coast from 2012 to 2021. And this started following the spike that we see in 2017 started following Hurricane Harvey, which, if y'all remember, big hurricane, lots of flooding. And while most bays saw an increase in alligator gar, Cedar Lake's catch, for whatever reason, skyrocketed. It may be because, you know, it's near that major river, the Brazos. There was a lot of flooding that happened from that river, but you would also expect to see it in Galveston Bay because that was right next to Houston. There was a lot of flooding there as well. So it's very interesting that we see this spike. This has been maintained through 2022. So this small area is kind of an ideal place to follow alligator gar around. So as you can tell from the map, we chose 13 receiver sites, and we implemented VR2TX receivers on either existing structure, such as pilings, or created our own structure using PVC poles, which was a whole trial and error process in and of itself. So from March to November of 2023, we tagged 54 fish. They were collected using TPWD resource sampling gill nets, which are left to soak overnight. And that's just our regular sampling that we do. And then also we use specialized 100-foot gar gill nets that typically used in rivers. We would soak those for an hour and kind of sit on them and, you know, see if they caught a fish. We would tag that fish. So a lot of you are probably wondering how we tagged these absolute units, especially with the scales. So it turns out they have a soft spot right underneath their pelvic fin. And so you can just make a small incision there, and we inserted V16 coded transmitter tags between the skin and muscle layer and sewed them up and sent them off on their merry little way. We also tagged the tail with a T-bar tag. In case the fish was caught by an angler, then they could call us and let us know. And we could take the fish out of the survey or mark it as a release. So our latest download was March of 2024, and the next download is actually being collected as we speak. So hopefully we'll start to be able to really get into that analysis. Speaking of analysis, the consolidation of the acoustic data we downloaded from the receivers was done using the packages Actel and RSP in R. Actel is a program designed to transform telemetry data to analyze animal migration and residency data. We primarily used it to acquire exploratory data about our fish's movements and consolidate the vast amounts of spatial and temporal data that those receivers produce. We also used RSP, which is a toolkit for the analysis of fine-scale movements in estuaries, and basically provide a more realistic estimation of space use. So basically what it does is it uses GIS to actually map the real land area, and it basically just gives you more accurate distances traveled. So from here, we plan on using various ecological modeling techniques. Still not 100% what we're doing with that. That's still a work in progress at the moment. But the goal is to input, you know, these biological information we get from the fish, the environmental data that we've been collecting at the stations, alongside this distance and movement data that we are getting from Actel and RSP. And we'll get into a little bit of that today. But again, what I'm showing is preliminary. So let's get into some of the results. Let's look at our basics. How far did the fish travel? How many of the fish actually were traveling? So out of our 54 tagged fish, 49 were detected by the receiver array, which I think was pretty good, all things considered. And out of those 49 fish, 35 made measurable movements through the array, meaning that they were detected on one receiver and picked up on another receiver. These movements varied greatly, as you can see from this figure. These are all the fish that made movements, how far they traveled. And the distance ranged from 1.4 to 78.5 miles, as far as how far the fish traveled. So there's a lot of variation between individuals. The remaining 14 fish that we included in the study were detected repeatedly but intermittently at the same receiver, so no measurable distance was calculated. These fish were determined as not dead, as these fish would go missing for a few days, up to a week, maybe even longer, before being detected at the same receiver again. If a fish sat on a receiver throughout the day and was pinging the receiver all day, every day, it was considered dead. Out of those 49 fish, 21 fish appeared to leave the array system as of March of 2024. So let's look at what this looks like spatially and through time. So here we have the seasonal differences in movements illustrated by the number of fish detections at each site. The size and colors of the bubble indicate the number of fish movements detected. So here we see most of the movement is occurring in the Cedar Lakes Marsh Complex, though there is some movement in the San Bernard River as well in the spring. And then movement in the rivers becomes basically non-existent in the summer, with the vast majority of detections occurring again at Cedar Lake. Once we look into fall and winter here, we start seeing movement shift from that Cedar Lakes marsh complex down there in the south up into the rivers. At first, both the San Bernard and the Brazos, but as winter rolls around, we see heavy activity in the Brazos River, which is that really, really long river. So it's possible that these fish are utilizing the marsh habitat throughout the warmer months of the year, foraging and move into deeper water during the winter, seeking potentially thermal refuge, maybe preparing for spring spawning, or maybe just following food resources. So this figure is another way to look at the same data. This is the overall movement of all fish in the study over space and time. We see the number of tags detected on the y-axis at any given location, which is differentiated by the colors at any given day, which is shown on the x-axis. So over here to the right, I've created a better legend that basically shows which general locations are represented by what color. It's a more concise version of that legend right there. Hopefully will make more sense to those that are unfamiliar with the study area. So what we see here is most of our tag detections is occurring in spring through fall, with the majority of those detections occurring in the Cedar Lakes Marsh Complex, represented by that turquoise blue. We do see some outward movement of fish leaving the area in the summer, either via the East Matagorda Bay to the south, which is that dark navy, and the San Bernard River, which is in that light green. And that makes sense, as these are the areas directly adjacent to the Cedar Lakes marsh. Then we see fish reaching the Brazos and all the way to Galveston Bay, which is to the north end of the array in the late summer into fall. And this indicates that these fish are highly mobile and are using the ICW to migrate between areas. Then we start seeing a transition in autumn, particularly in October onward into the winter months. Detections of fish start to go down significantly in general, especially in the Cedar Lakes Marsh Complex, but increase in the rivers, particularly in the Brazos River. And these patterns kind of show the same thing we see on the maps, and that's fish seem to be utilizing the marsh habitats at a more significant rate in spring and summer, and slowly transition to river systems in the fall and winter. Now, because of the extreme variance in distance traveled and the behavior between the fish, I went through and separated these two groups of fish based on a variety of parameters. So one of these groups is we called resident fish. These were fish that spent over 75% of their time in the Cedar Lakes Marsh complex where they were released and did not make significant movements. All remained in the area after the beginning of August. The other being mobile fish. These fish made significant movements throughout the array using the ICW, many of them leaving the array and either via the two rivers or through the intercoastal, although a few ended up back at the Cedar Lakes Marsh Complex. So one of the most significant differences between this group or these two groups was their size. Mobile fish were significantly, though not extremely, larger than the resident group. This trend is further shown when comparing distance traveled in kilometers to the total length, showing a positive correlation between length and the distance the fish traveled. This could just indicate that smaller or younger fish tend to stick to estuarine areas and become more transient as they age or as they increase in size. And this could be due to a variety of factors, including, a need to spawn, following resources, and it is interesting. And it's something we want to kind of look at going forward as we tag more fish. So this is the last thing that I've been looking at. This is comparing movement to environmental factors. So when we look at environmental factors associated with season, we see these trends start to be more fully teased apart. So this area is prone to extreme swings in temperature, salinity, and dissolved oxygen. One, because all these bays are very shallow, they have significant freshwater inflow, and it gets very hot in summer. So it creates these really extreme conditions. So what I did here is take these three variables of importance, temperature, salinity, and dissolved oxygen, and put them alongside the total distance traveled by all fish in kilometers as a proxy for fish movement. So looking at salinity here versus average distance traveled by all the fish from April to February, we see that movement increased as salinity decreased. In addition, when salinity increased above 30 parts per thousand in the summer, movement slows almost entirely. Many of these fish were just residing in Cedar Lakes. Some had moved out of the area. And it's possible that some fish may be avoiding some of these extreme conditions, or some fish may not be moving because of the physiological stress of these conditions. In addition, when you see that movement start to pick up again in the fall and winter as salinity decreases, it could be that freshwater flow could actually trigger movement in the species. When we look at temperature, we see a similar trigger for movement as temperature begins rising towards those high summer temperatures. Mobile fish have maybe left the system, or fish are just residing in place. And in the winter, we start to see a decline and overall fish movement increases towards the rivers, potentially to use them as a thermal refuge from the shallow waters of the marsh, or to follow resources. So we're seeing similar trends here as well. When we add in dissolved oxygen, we see dissolved oxygen start to decrease. In this case, it was due to high temperatures, drought, and red tide that summer. Again, we see fish either leaving the system or slowing movement entirely. And when dissolved oxygen returns to more tolerable levels, movement increases. Really, what's likely happening is that these three variables are working in tandem to trigger fish movement and may be more indicative of a seasonal driver of movement. However, we would like additional data before we do more complex ecological modeling. Right now, we only have a year of data. There's a lot of variation from year to year, especially in an extreme region like the Texas Gulf Coast. For example, we had drought the last 2 years, but the following year, this year, we may have an overabundance of rain. In addition, this year, from the data that I'm showing you now, we had a significant red tide event. They did not have a significant red tide event this year. So collecting more annual data will help us determine if this really is a seasonal movement trend or if this is just interannual variability. So our preliminary data does allow us to draw a few conclusions moving forward. Clearly, gar regularly access both estuarine and river mouth habitats throughout the year and are using the ICW as a movement corridor, which is really cool. Though clearly movement behavior between individual fish varies greatly. Movement seems to be timed to certain water conditions at certain times of year, though physical characteristics of the individual fish may also contribute to how far the individual moves. So based on our preliminary data, fish use estuarine habitat in the summertime and then move towards the rivers, particularly the Brazos in the fall and winter. Larger fish were also more motivated to travel farther distances. And this could hint that these fish may be more motivated to enter rivers for spawning in the spring. However, a majority of these fish we tagged were most likely not reproductively mature. So I think it's more likely that these fish are motivated by ideal conditions, taking advantage of either the foraging abundance in the estuary in the spring, maybe leaving in the summer when conditions get too intense, or limiting their movement, and then entering deeper water in the rivers when temperatures start decreasing, or potentially still following that food resource. So our office put in a grant submission for future funding with a couple of main goals moving forward. We are continuing to collect otoliths from coastal gar in several bay systems to create an age and growth analysis for coastal alligator gar. They will also be tagging an additional 25 fish in the Cedar Lakes array, as well as continuing to download our data. The next set of data is being collected as we speak. We have also proposed an array expansion into East Matagorda Bay, which is directly south of our current array, where we hope to tag 50 additional fish and establish an array of approximately 18 loggers down there. And besides just expanding what we know about gar movement, this would open the doors to developing projects involving other highly mobile predatory fish, such as sharks or red drum. And then overall, this project hopes to expand the Texas Coastal iTag network, which at the moment fully connects Sabine Lake through Cedar Lakes. And we also plan on publishing this work, which is something I will likely be working on this winter to get the data collected from this year. So I'd like to thank a few people. Number one, our technicians down at Perry R. Bass, Kevin Willis and Billy Sprouse, who helped me significantly with array implementation, maintenance, and fish tagging, and they are continuing to do that now as we speak, even though I'm all the way up here in Massachusetts now. Our project partners are Carrie Gelpy and Shambu Paudel from the Sabine Lake office. Without them, we would not have been able to get the telemetry aspect of this project up and running as quickly as we did. Dan Doherty from Part of the Hills Fishery Science Center, that's our inland component. And he is kind of the alligator gar expert in Texas and has been pretty useful for this project. And then our funding was from Gulf States Marine Fisheries Commission from the SURF grant program. And with that, I will take any questions.
Thanks, Jillian. Do you have any questions for Jillian? Either unmute yourself or put something in the chat.
I will ask a question. It's Tracy. Jillian, you mentioned that you thought a lot of the fish that you were tagging were likely not mature yet. Have you seen anything to suggest that there's maybe a maturing aspect to who can hang out in those higher salinity areas and then who has to go up the river. So like maybe the reproductive fish are less tolerant of those higher salinities.
So from the research that has already been done, there hasn't really been a delineation between like the juvenile, quote unquote, adult large fish and then like the actually reproductively mature fish. So we're hoping that like with creating that age structure and then using length as an identifier of, sexual maturity with this project, we may, be able to kind of suss that out.
I have a question. It was interesting seeing that jump up in 2017 in the
Yes.
CPUE. Do you think that had to do with the hurricane? Was there a lot of water that may have pushed individuals down from the river systems and stuff into the estuaries?
Yeah. So I wasn't actually there during Harvey. I thankfully got to avoid all that by coming in 2019. But from what folks have told me, there was an extreme amount of flooding in the area that we studied, as well as Houston. I mean, entire interstates were underwater. And just from personal observation, whenever we get a lot of rain, the locks on the Brazos River tend to overflow. We get a lot of trees getting washed down river and into the estuary. So yes, there's a lot of flooding that definitely happened around Harvey. And I do think that that's part of the reason why alligator gar are far more prevalent in the area. Now, as to why they're prevalent there and not in, Galveston Bay near Houston, I'm not really so sure about why that's the case. It might have something to do with just the variety of habitat available to these fish in Cedar Lakes, both the estuary and the river. So yeah, Harvey definitely had, I think, an impact on why we see more gar. Yeah.
Adrian Jordan has a comment. Looks like overlapping numbers of individuals over times of the year. Any thoughts about prey species and competition?
Yeah, so as far as prey species are concerned, alligator gar will eat anything and everything. I mean, they're like catfish, in that like they've found birds and small mammals in them. So they're not really targeting specific prey as far as, as we understand it. It's more that they're following that prey abundance, which does change throughout the year. And as far as species competition, like between other predators, they don't really have competition. We do get those big bull sharks, but for the most part, I mean, alligator gar are absolutely dominating. So I don't think competition plays a lot into that. As far as competition between individuals, yes, that might be why we see some of the larger individuals moving to different areas.
He says, cool, thanks. Any other questions for Jillian?
Or suggestions. If you have any suggestions on how to analyze this data, I'm all ears.
You should talk to Micah Dean, who's and Ben Gahagan, who've been doing all this work for striped bass and cod and stuff like that. So I could probably give you some suggestions.
Yeah, it's definitely more comparable, I think, to your more riverine species as well. Yeah.
Well, I don't see any hands up or other questions, so just wanted to thank Jillian for her time. Adrian also said, talk to Ted Castro Santos at USGS, does work on freshwater movements.
All right, thank you.
Okay, that looks like it. So I just want to thank Jillian again. Thanks for her time, and thanks everyone for attending. So the next seminar will be in January after the holidays. So hope to see you then. And Jillian, there's some clapping going on in the
Thanks.
things.
And I apologize for the delay. I still don't know what was going on. We had 3 people working on it. It was nuts. So I super duper apologize.
All right. See you, everyone. Thanks again.